I'd like to introduce our first speaker, who is Associate Professor Prue, and she's providing an update on the Breast Cancer Trials Research Program. Prue is the head of medical oncology in the breast service at the Peter MacCallum Cancer Centre and a consultant medical oncologist at St Vincent's Hospital in Melbourne. Prue is also the chair of the Breast Cancer Trials Scientific Advisory Committee. Hi, Prue.
Hello. So thank you for taking the time to join us this evening, and I've had the privilege of chairing the Scientific Advisory Committee of the Breast Cancer Trials Group since 2017, and I wanted to give you a few insights into where we're at with our trials currently. So one point I wanted to make is that there's never a good time to be diagnosed with breast cancer. However, it's also true that there has never been a better time than now to be diagnosed with breast cancer. And why do I say that? I say that because survival for breast cancer has continued to improve, and the improvement is actually mainly due to the routine use of treatments that were previously tested in clinical trials. And as the decades go on, the survival is continuing to improve as we implement treatments that we've tested, various chemotherapy regimens, different types of hormone approaches, HER2-targeted therapies for those with HER2-positive cancers, radiation where appropriate, and also some new biologic therapies. The other thing that's important to remember is that clinical trial participants are the very first to access new promising treatments. Now, what's different about clinical trials in breast cancer in 2020 compared to perhaps a few decades ago? So currently our trials are not just about adding a new treatment. Nowadays when we do a trial, we're doing very detailed studies of the participants' blood and tumour samples and doing some very complex laboratory analyses of these samples where consent is provided to try to understand better our treatments and how to target them. In our trials, we're really aiming to target new treatments to those participants or patients who are most likely to benefit. So we've moved from a one-size-fits-all type of trial to more targeted approaches in many of our trials. And when we're thinking about trials currently, we're really aiming to tailor treatments so that we can actually optimise the individual patient outcomes. And in a trial setting currently, that might translate into treatment escalation, meaning more treatment for a particular patient, or it might lead to a de-escalation trial where actually that patient might be offered a trial with the possibility of less treatment. And our goal in our trials really though remains the same. We're aiming to improve the length and quality of life of those affected by breast cancer and also to prevent breast cancer. So how do we decide what to study? So it's not just me deciding, which is the good news. So we do have a scientific advisory committee and we have 26 members of this committee, and these members are made up of breast cancer researchers from right across Australia and also from New Zealand. And the members of this committee have quite a range of different expertise. So we have surgical specialists, medical oncologists, radiation oncologists. We also have translational researchers. And these, if you're not familiar with that term, it refers to an oncologist who also runs a research laboratory. And those people are very helpful because it means that anything that we learn from a trial can be analysed in more detail, and then they can bring us ideas back to us for new trials. Importantly, we have consumer advisory panel members as part of our scientific advisory committee, and they're integral in looking at all the things that we're thinking of studying. We have a psychologist involved in our committee, we have a statistician, a pathologist, and a COO. And apart from this committee, we actually call on a range of our other members, and including our full consumer advisory panel, to review proposed trials and trial concepts. So like everybody else, we had to hold our committee meeting by Zoom recently for the first time, and despite the fact that we were from many different cities and even from New Zealand, we managed to do this for a half day recently in July. Now, what are the areas that we have been researching recently, either current trials or recent trials? So we're interested in preventing breast cancer, and we've got a current trial called BRCA-P, which I'll talk about a little bit in a moment. We're interested in breast cancer imaging, and we've got a current trial looking at the role of MRI in the workup of breast cancer when patients are going through the diagnostic process. We have looked at ductal cancer in situ or precancerous changes, and we have a trial called IBIS-2, which patients are still being followed on. We've had surgical trials, and a recent example of that, which is currently active, is the POSNOC trial. We have a local therapy trial which recently completed recruitment, and we're expecting to have first results next year, and that's the PROSPECT trial. We have a radiation trial currently looking at whether some patients could avoid radiation, and that's called the EXPERT trial. We have trials for patients with early breast cancer, what we call adjuvant trials. An example of that, which we recruited a large number of women to, was the PALLAS trial. We do neoadjuvant trials, and that's trials where we're testing giving treatments prior to surgery, often drug treatments prior to surgery, and a couple of examples of these are the CHARIOT trial and the NEON trial, which are both in triple-negative breast cancer. We also do trials in the post-neoadjuvant space, and I know that's a big mouthful and may not mean anything to you, but that refers to patients who have residual cancer at surgery after having undergone preoperative drug therapy. We have trials in the metastatic situation, and 3 examples of those are listed here. We also have trials or research studies specifically looking at patient care issues, and examples of these would be developing decision aids to help patients make decisions about components of their care. We're interested in anemia that might be experienced in chemotherapy, interested in problems related to vaginal side effects of breast cancer treatment, nausea, sleeping difficulties. We also do translational research, and one of the areas that's been of interest to some of our team is looking at tumour-infiltrating lymphocytes, also called TILs for short, which is found to be an important marker of future prognosis in women with triple-negative early breast cancer. So just to give you an example of some of the trials, this is the BRCA-P trial that I referred to earlier, and this is a prevention trial, but it's in a very specific patient population, and it refers to the patients that have a BRCA1 gene mutation. So it's not for all women, but women who carry a particular genetic predisposition with a high risk of developing breast cancer. And in this trial, women will be randomised to either receive or to not receive a drug called denosumab, which is in other situations used to strengthen bones. But in this particular trial, based on laboratory research from one of our translational researchers, we're looking to try and prevent breast cancer in these women at high risk. An example of a trial in metastatic disease that we're currently participating in and conducting is the DIAMOND trial, and this is an exciting trial. It's a study testing immunotherapy and combining immunotherapy with the standard HER2 treatment of trastuzumab in women who have metastatic HER2 positive disease that has increased after previous therapy. And in this trial, we're trying to look at an approach where we're not actually giving chemotherapy, but combining immunotherapy with trastuzumab and trying to look if we can get disease control. And then I talked about de-escalation trials, where we're thinking about identifying patients with tumours who might have a good outcome despite reducing treatment. So a de-escalation trial might refer to a smaller operation, it might be reducing drug therapy, or it might be avoiding radiation. And an important example of that is our current EXPERT trial. Now, not all breast cancers are biologically equal, and when oncologists talk about breast cancer, we have a shorthand that we use. We refer to luminal breast cancers, which are hormone receptor positive breast cancers, and then we talk about triple negative and HER2 positive. And within luminal breast cancer, there are a couple of different types of luminal breast cancer. There are those we think of as luminal A, which tend to have a slow growth, and there are luminal B, which can have a faster growth. And for women who have what we call a luminal A breast cancer, a slowly growing hormone receptor positive tumour, we're looking at studying those in a trial called the EXPERT trial. And in general, when women have breast conserving surgery, it's standard to offer radiation to the conserved breast. But in this trial, we're looking at women who are at least 50 years of age with a breast cancer that's up to 2 centimetres in size and a tumour that does not look aggressive under the microscope. And the tumour does need to be hormone receptor positive and HER2 negative. And they're planning to get hormone therapy. And if women agree to participate in this trial, we would then send a small sample of their tumour that's been removed off to a specialised laboratory to get a multi-gene assay called a PAM50 assay. And if this assay confirms that they do in fact have a luminal A subtype of breast cancer and their risk of recurrence score on this particular test is not considered high, then those women can be randomised to either receive hormone therapy with standard radiation to the breast or hormone therapy without radiation. And these women will be followed for 10 years. So what's unique about the EXPERT trial is it's the only trial that's randomised in the world that is testing radiation to the conserved breast versus no radiation using a multi-gene assay test in early breast cancer. So these multi-gene assay tests have been used sometimes to help with chemotherapy decisions, but we're trying to test whether it could be helpful in making radiotherapy decisions. Uniquely, our group designed this trial, developed it, and we're coordinating this trial across multiple continents around the world. And we believe this trial does have significant potential to change future breast cancer treatment. So that's a bit of a whistle-stop tour through a little bit of what we're doing in breast cancer trials, and I'll be happy to answer any questions.
Thank you for that, Prue. That was a really good overview. And thank you for mentioning the BRCA-P clinical trial as well, because we currently have a campaign out in the community that people may be seeing on Facebook or on digital advertising or in media. If you want to learn more about the BRCA-P clinical trial, have a look at breastolution.com.au. And Prue, I've got a couple of questions for you that have come through. One question is from Joe, and Joe asks, how are patients chosen for clinical trials?
Right, so I think with a clinical trial, it's really, we're looking at it from two different perspectives. We're wanting to find, with any patient, we're hoping to find a trial that might be suitable for that patient, that they could be offered. So we're wanting a trial suitable for the patient, but we're also wanting the patient to be suitable for the trial. So it's kind of a two-pronged approach. So in thinking about a patient that's suitable for the trial, we'll be thinking about their general health status, what previous health issues they've had, their age group, their specific subtype of cancer, because breast cancer nowadays is not all considered to be one type of breast cancer. It's quite nuanced. And so we're thinking about many different factors before we decide whether a trial would be appropriate to offer that patient and whether that patient would be suitable for that trial. And so, but certainly for researchers such as breast cancer trials researchers, we are very focused on making trials available. And one of the nice things about our network, because we have so many centres across Australia and New Zealand, that if a particular trial from our network is not open at a particular site, we actually provide information that allows other, the patient potentially could, with their permission, could be referred to another site to participate in the trial at a nearby site if they were interested. So we do try to maximise the opportunities for patients to participate in trials according to their health status and their type of cancer. All right. And their preferences.
Thank you, yes.
Most importantly.
Well, I wanted to ask you a final question too. So with this question was sent through by Nicole, and there's quite a few people who have asked the same question actually. So Nicole asks, what is the latest in research and trials on triple-negative breast cancer?
Well, there's a lot happening in the triple-negative breast cancer space, and I probably can't cover it all this evening, but suffice it to say that I referred briefly to TILs, the tumour-infiltrating lymphocytes, and that's something nowadays that we're thinking that increasingly some pathologists might start to report in pathology reports, which might give some extra clues about prognosis in early triple-negative breast cancer over and above tumour size and lymph node status. And that information was derived from our previous breast cancer trials. But we are now testing some new approaches in triple-negative breast cancer, looking at whether we can harness the immune system better in conjunction with chemotherapy to try to obtain better cancer tumour shrinkage, often prior to surgery. And in triple-negative breast cancer, increasingly we are thinking about trying to treat the cancer before surgery so that we can get a sense of how responsive it is to those treatments.
Well,
In the metastatic space, again, immunotherapy is becoming an important thing to consider in selected patients.
Yeah. Well, thank you so much, Prue. Unfortunately, that's all we've got time for on your section tonight. We've got to keep rolling along, but thank you so much for your presentation.
My pleasure.